You're listening to the Effective Statistician Podcast, the weekly podcast with Alexander Schacht, myself, and Benjamin Pieske, designed to help you reach your potential, lead great science, and serve patients without becoming overwhelmed by work. Today I'm talking about a design that I have never talked about before: crossover, the benefits and the problems around it. And I'm talking about this not by myself, but I'm talking with Markus Savli. He is a great statistician, very passionate, easy to work with, and runs his own company out of Switzerland. So stay tuned for this really nice discussion about this very insightful and interesting design. This week, as this podcast episode becomes live, we will have another webinar. This webinar will be about resolving conflicts, and we'll talk a lot about conflict and negotiation and all these kind of different things. I know this conflict is an almost always for sure to come up topic in all the different leadership programs. And having conflict with physicians, with programmers, with CROs, with upper management, all these kind of different things, it happens again and again and again. And it can be so frustrating. I can tell you that I have also suffered a lot from bad conflict. And I'm not talking just about the conflict where people kind of, hate each other and, write nasty emails, all these kind of different things. But also about the passive-aggressive conflict where you're not included in certain informations and things like this. This can be as distracting, as demotivating, and really even leading to burnout. So register for this upcoming webinar on November 1st. It will happen in the European afternoon, so in the US morning, and this week is a little bit off with all the time zone changes, so have a look into the Effective Statistician homepage, check for this webinar and register now. You can also find the link to it in the show notes. So now stay tuned for this really good podcast episode. By the way, there's one other thing I need to talk about. Currently, there's a call for the abstracts for the PSI conference in Amsterdam in 2024. This conference is one of the absolute must-go-to conferences if you're in Europe, and so come there, submit your abstract. Although it's quite a fierce competition there, do it. That will increase your likelihood for getting approval by your supervisors, and come there. I'll be in Amsterdam for sure. So go to the PSI homepage at psiweb.org so you can learn more about all the other PSI activities, but for sure to check out the timeline, which is end of the month, end of November, so that you submit an abstract. And of course, this podcast is run in association with PSI. Welcome to another episode of The Effective Statistician, and today I'm super excited to talk about a topic that I haven't touched on in over 300 episodes, and I can't believe it. It is such a classical design. Although I need to confess, I've personally never worked on this because, we'll get into the reasons later on. So, and for this, I have Markus here. Hi, Markus. How are you doing?
Hi, Alexander. Great. I'm doing fine. Thank you for the invitation.
Nice to have you. So let's start. Introduce yourself. What are you doing? What's your company doing?
Markus Savli is my name. I'm biostatistician. By training, I have a background in biomedical engineering and mathematics, as well as epidemiology. I work at and the founder of Biostatistic and Consulting. We are a consulting office in Zurich, in Switzerland. And as the name already indicates, we offer biometric services for pharmaceutical and biotech, medtech companies. But furthermore, for universities, for example, we can do scientific writing, like meta-analysis or similar services, programming, dashboards, and so on, right?
With your background that is so broad, much broader than most statisticians come with, who have spent their whole career only in clinical trials. So biomedical engineering, programming, really kind of a broad data science background. This is pretty cool.
Yeah, thank you.
So today we will talk about crossover studies. And you recently published one, and we'll go into this a little bit later. But let's, in simple terms, what's a crossover study?
A crossover study, in simple terms, is a longitudinal study where subjects receive multiple treatments in a sequence. This can be an observational study, where it's less controlled, and this is what we are more interested in the medical or in the scientific area, to have a controlled experiment. So then it's randomized usually, and it's balanced. That means the same number of subjects receive the same number of treatments and the same number of periods.
In the most simple case, if we have two treatments, treatment A and treatment B, half of the patients first get treatment A and then treatment B, and the other half gets first treatment B and then treatment A. So we have two treatments and two periods.
Right. This is, you explained already, the randomized part of it, right? So you don't start with the entire number of patients with treatment A and then cross over to treatment B. You randomize them. That's correct.
So that first kind of appears like, looks like an unusual design. Why not just kind of compare, A and B, have the first period, that's it. Why do the second period?
This comes with certain properties. First, you can consider when you take a subject and have him get him one treatment and then the other, or the placebo treatment, the same subject serve as its own control. And the advantage of this is that you can influence the confounding, right, in a better way than you can do in a randomized or two different groups, right, than in a parallel design, for example, where it's a bit less uncontrolled. And given these circumstances, it gets along with a higher statistical power. And higher power means that you can get along with fewer subjects. And this is a matter of cost in the end, right?
Yeah, so especially if you have high variability between the different subjects, that is clear that, the within subject control gives you much more additional comparisons, because you don't just compare treatment A and treatment B between the different subjects in period A and period B, actually, but also you compare period A to the first period to the second period, and also have these comparisons. So cool, very good. So you have, instead of just one comparison, A versus B at the beginning, you have basically four comparisons. A versus B in the first period, A versus B in the second period, and A versus B in both groups, so to say. Both do you call them sequences or groups? How would you call that? Patient.
Yeah, sequence. Yeah, sequence is good, right? Period. In the ideal world, there should be no difference actually. When you start with A and cross over to B, or you start with B and cross over to A, this is actually what we want to find out in this study, whether this is really the case.
Yeah, I think that is one of the assumptions that you need to make, isn't it? So it does matter whether you first go to B and then to A versus A and then to B. So the treatment difference between A and B depends on the period. Why is that a problem?
Yeah, you don't want to experience this in your study, then you really are a bit in a mess. For the point is, for some study designs, there's actually no other option than do a crossover design. For example, bioequivalent studies, right, where the guidelines actually tells you that you have to do this, and then you need to control for these effects. We talk about like an order effect, what you mentioned before. This is when the sequence may affect the outcome. An example could be that, let's say, the first treatment produces more side effects or adverse effects, right, in the patient. And then in the second later treatment, where there is less side effects, but the patient is more susceptible or more kind of framed for the side effects and is perhaps reporting more adverse effects than they actually are, right? So this is what we don't want to have, and that's why we do randomization, where we can check, is it from A to B or from B to A, right? Is there a difference, right? And if it should be balanced, actually, there should be no difference between these sequences.
This is a carryover effect.
No, this is the order effect. The carryover effect is another effect. It is where the treatment itself lasts long into the next phase. So it confounds the effect of the next phase. In a good crossover design, there is sufficient time between the treatment effects or treatment periods, and we call it a washout phase, so that there is no interference. And they say, well, in the guidelines, say more or less 5 half-lives, right? It should be sufficient, right? And you need to check this in your data, right? It's the case, right?
So that already has a couple of different implications. So first, it only works for treatments that have reasonably short half-lives. Yeah, so if you have a half-life of several months or something like this, it becomes a very, very long study. The second is, of course, that you have some kind of chronic stable disease. Yeah, if you cure the patients with your treatment, that's probably not the right way, because then you will always have a carryover effect. So it needs to be some kind of symptomatic treatment. And yeah, so you need to have this kind of, you need to more or less get back to the first baseline. Yeah, so basically you have 2 baselines, isn't it, in the settings, and you have a baseline for each period.
That's right. You mentioned a couple of points, right? That this crossover is not suitable for all situations. And when you cure the patient in the first treatment phase, the baseline for the second phase is different, of course, and there is no more improvement for the patient in the second phase. So the second phase will be worse in the end in the calculation, but then the study design is not appropriate for that. But the biostatistician should know about it and about the effect. So in the curative situation is perhaps not the ideal study design, but for the chronic ones where you apply certain treatments and then the patient benefits during this treatment phase in a certain way. But then when you stop this treatment and the baseline returns to its original level, then you have a good situation where you can independently check both treatments. It could be the active verum and the placebo situation, right? Or in the case of bioequivalence, where you apply kind of a single tablet or a single application of a drug, right, where you check the pharmacokinetics. So after a certain half-lives, the pharmacokinetics returns to its baseline. This is the case when, bioequivalence has its value because, for example, a company decides to change its formulation from a suspension to a tablet or vice versa, right? The properties are similar according to their production, but still the government and the authorities require that you need to check this. And then with a single formulation, right, you do the pharmacokinetics, blood AUC, and so on, and then you compare this. So in that situation, the baseline is established, and you can go ahead with it.
Okay, awesome. Let's go into the problem. Is very simple: 2 treatments. What happens if we have multiple treatments? So let's say 3 treatments. let's make one step further. Do we then always have also 3 sequences? So you have basically treatment A, B, and C. So you have A, B, C, A, C, B, and all these kind of different combinations.
Yeah, the simple design is the 2-period 2-sequence crossover, right? This is the most simple when you have let's say the active and the placebo or 2 different ones. But when you go for 3 ones, it could be different dosages or like this. Then it's not 3, you get 6 treatment sequences. Because of this, you want to randomize all the sequences and want to check and avoid actually any, or you don't want to miss any effect, right, for a certain sequence. So you need to randomize of these 6 treatment sequences, right? And to assign the patients, you do a block design. And what we do is that we have a complete design. That means every patient finishes all the 3 sequences, right? And we have a couple of patients in each of these sequences, right?
So the study gets longer, and of course you need a certain number of patients for each of these 6 sequences. Okay. I guess that's probably the maximum that you would use. Going to 4 or 5, it gets more and more complex. Or have you seen crossover studies with even more treatments?
I myself was not involved in such studies, but the 3 periods is not so uncommon, I would say. 4 or 5, I mean, theoretically should work, but I cannot remember when I saw one. Actually, I haven't paid attention to these higher sequences, but as you said, is for sure more complicated and you need to pay attention to the details then, right? Yeah, but I think theoretically it should work.
The studies get longer and longer and bigger. And of course, you increase the chance of something going wrong, like dropouts. So that is what was one of my first, questions when I saw this design. Okay, how do you analyze patients that drop out between the 2 sequences, 2 periods, for example?
Yeah, this is always the case when you have a longitudinal study design what often happens and is unfortunate for the study design and for the analysis in the guidelines. When we go for let's say for the bioequivalence guidelines of the FDA, of the EMA, they require a minimum of around 12 patients, right? And so when you have around 12, 18 patients, right, they prohibit actually any extra patients, right? When you miss one and you just load another one into the study. No, this is not allowed. So, but on the other hand, they say, or they offer the option that you can account for dropouts. When at the beginning of the study, before you start, right, it is likely, or you assume, there will be kind of a dropouts because of this peculiar treatment, what you apply to them. You can account for these dropouts and, add this 10, 15, 20%, whatever patients at the beginning, right? So that when they drop out during the study, that you have still a sufficient high number of patients. Because when one period is missing, then you don't have this comparison, right? This is missing actually, yeah. And this is what we do. We just account for the dropouts.
If the patient drops out, you wouldn't consider them in the analysis because he doesn't have all the information?
Right, that's a problem. And that's why we have this ITT and per protocol options, right, and sensitivity analysis options, right? But when you do a kind of a mean comparison, right, between the 2 treatments, then it is difficult, right? Because you don't have one. And it's not like in a parallel design. Okay, you can make the mean in the one group with 1 or 2 or X patients more than in the other one. But in the parallel design, where you want to make use of different statistical methods, right, the higher power, then this one is missing, right?
Okay, so it's a much bigger problem compared to the simple parallel group design. Okay, very good. Let's go back to the studies that I mentioned at the beginning that you recently published and tell us a little bit about this crossover study because that actually had 3 different treatments.
Correct. That was not a bioequivalence study. It was a non-inferiority study in the field of allergy. It was a medical device product, kind of an over-the-counter drug, so less stringent than other pharmaceuticals. And the sponsor wanted to compare his product to an already marketed competitor. And in the allergy area of field, the patients are very sensitive, right, to the environment and also to the treatment. So the treatment effect is small and highly variable, right? Mm-hmm. And that makes it really difficult to assess these treatment effects. And we don't say drug. The over-the-counter is a kind of a nose spray, right? Mm-hmm. It's applied into the nose to relieve certain allergic effects, right? Or the idea was to check what is the potential for the allergic relief, to get this relief. And they wanted to compare it to this other drug and also to kind of a non-treatment. So the idea is, okay, if this is so highly variable, is this pure chance or is this how does the patient fare off when he's doing nothing? And how do they do this allergic study or check this treatment effect? They usually do it in winter time. Why? Because then there is this pollen. There's no pollen season, so it's during the day. The temperature is cold and there is no other grasses or pollen around or less. And then they have the patient in a controlled environmental chamber. That is, you can say, kind of a small room in the laboratory, and the patient sits there, and this room is air conditioned, it is temperature controlled, it is humidity controlled, and whatever other environmental parameter controlled. And via the air condition, they expose the patient with a certain set of pollen and other allergens. Okay. And the patient sits there, and then the exposure lasts from 3 to 6 hours, depending on the study. And the patient sits there, and then he's there and
gets the allergy.
Gets the allergy, and then every 15 minutes he has to report his symptoms like sneezing, itchy nose, watery eyes, respiratory, whatever. There's a list of symptoms what he has to report, and then is done. And then after a washout period, he returns for the next one and so on, right?
Okay. Wow. So highly controlled environment in which you can also control the exposure. Okay, that's really, really interesting. So the exposure in terms of the allergens, not the exposure in terms of the treatment, which you can control as well.
It goes even that far. The way the tissues, right? When you have a running nose and they blow the nose, they collect the tissues and weigh the amount of what came out of the nose, right?
Wow. Yeah. Okay. That's really an interesting lab setting. Yeah. And a really great setting for these types of studies because you have these repeatable conditions. People in winter get out of the chamber again. They don't have any exposure to allergens anymore, get back to normal. You can have a washout period. And these are really, really good conditions for these crossover studies. And it's really good that you have these golden standard, so to say, approach here with active control and also a placebo control. So you can have all these things that you very often need for a non-inferiority study, because very often your non-inferiority margin depends on the difference between active and active and placebo. Very good. Awesome. We'll put the link to the study also into the show notes so you can have a look there. Where can people find you, Markus?
That's very easy. I'm, of course, on LinkedIn. That's one option. You can connect to me on LinkedIn and drop me a message, or to take a look at my website and see what else we are doing, the team, and by email. That's the traditional, old, but still working way.
Yeah, we'll put links to these kind of different things into the show notes so you can very, very easily connect to Markus. A couple of interesting things. Markus sits in Switzerland, and sometimes there's a lot of concern in terms of Swiss people about Swiss data not leaving the country. So if you need to work with, a Swiss biotech company or Swiss hospital or these kind of areas, this is very often a concern. And then having a Swiss consultant there that can do all these kind of different things without the data leaving the country, then for sure reach out to Markus. And then, of course, well, although he is from Switzerland, he actually speaks German. Also for all kind of German training, German HTA submissions, all these kind of different things. If you belong to the part of the world where your mother tongue is not German, which, yeah, is the biggest part of the world, and you need to have someone that is a good German-speaking statistician, I also highly recommend you reach out to Markus. Thanks so much, Markus. Is there any final things that you would like to give to the listener?
I can add Spanish to the options, language options.
Okay, very good. So we talked about crossover studies, the benefits, the problems compared to traditional designs. Have a look into this and increase your knowledge around these things. And for sure, connect to Markus. It's always great to improve your network, to strengthen your network. I always recommend this to people because your network is your net worth very often. Yeah, the more people you're connected to, the more and easier you will find maybe new colleagues, new partners, new employers in the future. So connect with Markus. If you haven't connected with me already, of course, connect with me as well. Thanks so much, Markus, for being on the show.
It was a pleasure. Thank you, Alexander, for inviting.
Don't forget to register for the upcoming negotiation and conflict resolution webinar that happens on November 1st, 2024. If you're listening later to this, you can still find it on the library. Go to theeffectivestatistician.com and check out the library where we have all the recordings. But I really urge you to register for it now so that you can get to it and really ask your questions and have all the discussions. That would be awesome. This show was created in association with PSI. Thanks to Rain and her team at VVS who helped with the show in the background. And thank you for listening. Reach your potential, lead great science, and serve patients. Just be an effective statistician.